And we're live.
Good morning,
everybody.
Say hi, Owen.
Hi.
Guest speaker. You want to say hi and then you're going to go eat breakfast?
Hi.
Okay, say see you later.
See you later.
All right. So here we go. I'm going to do that mute thing, Lisa, and then you can fix it. First of all, thank you all for coming, the people that are here. You didn't have to be here. Obviously, the school said that. But here's what we say. This is stuff you're going to have to do anyway. When we go back, if we go back in 10 days, we're going to use the exact same stuff we're using now for the people that aren't doing it now, and they're going to work through that. So it's really to your advantage to do it now. I think it provides a sense of normalcy for you to do it now and for us. Obviously, if you don't have to do it, we don't have to do it, but we're doing it because we want to. We want to be here with you. So really, I think really important because, guys, like the learning continues. Like the world's kind of messed up. Things are weird. 10 days off school. And who knows if that 10 days is really going to be 10 days or not. When you look at what's happening around the world, it could be something else. So what we're thinking is we're getting ahead of the curve. Instead of sitting at home and doing nothing, we're going to get ahead of the curve. We're going to use the time, and we'll reward you for that when we get back. All right? That's where we're at. So today we are talking about replication, and I have to do one thing before we get into that. I have to send out to you the attendance sheet. So there's a Google Doc, Google form coming out in the chat. Please click on that. That's the attendance sheet for today. Just fill that out real quick. And at this point, I'm going to turn it over to Mrs. Smith, and we're going to start talking about DNA.
Okay, so if you're my student, you know I love to do daily questions. So I thought we'd continue that and do daily questions. You're going to type right in the side if you know the answer. This should be a straightforward, easy one for you. What are the three parts of a nucleotide? I'm going to give you a second to think about what the three parts of a nucleotide are, and you can type them over on the side. Let's see who is going to be first. And I'm sorry if you're my student, I realized that I had the link locked, so I did send it out. But three parts of a nucleotide: nitrogen base, sugar, and phosphate group. Nice job. Daniel Corbin was in first. Oh, wait, no. Wait a second. He popped up, and then I see Haley had some of it. I see Erica. Nice job. Okay, a bunch of people popping in. That's great. Okay, so absolutely, you know, we're going to have, and Suter, if you want to flip over for a second to show them, we're going to have the three parts of a nucleotide. You have the phosphate group. You can see in the blue, okay, you have the pentose sugar for DNA. That is a deoxyribose sugar. And then you have a nitrogenous base, which it can have one ring or two rings. So which one has one ring and which one has two rings? Think about the little trick we talked about yesterday. And you can type that on the side too. We'll see who knows how many rings are in which first. Okay, so the other thing we're going to talk about is a little bit more with the directionality with DNA today. Okay, so I'm going to flip over to my whiteboard. You know, I've always wanted a whiteboard in my room. Now I have one at home. So when we talk about directionality, okay, what we're talking about is basically the position of DNA. And Suter, are you going to make me full screen so that's a little bit clearer?
I don't have an answer for that.
Oh,
is there something else I can help with?
My phone. I said Suter, not Siri. All right. So when we talk about a DNA and we talk about having our nucleotide, okay, we have our pentose sugar. Okay, that's going to be connected to our base. Okay, one of our nitrogenous bases, and then that's going to be connected to a phosphate group. So I'm going to kind of put it like this, and I left a blank there on purpose. At the very point, Mr. Shooter talked yesterday about this being that oxygen. Okay, and then what I want you to think about is that if you go around to each side and kind of think about that glucose molecule that you made, that's a sugar molecule too. You put the oxygen at the top, the red oxygen, and then you had the black carbons. And when you did the black carbons, you put one here You put one here, put one here, put one here. And then remember you had that one that sticked up with the 3 hydrogen molecules attached to it. So the same way, the ribose sugar or deoxyribose sugar here also has a carbon that sticks up. So what we like to do is we like to count the carbons. So starting at the oxygen, going clockwise, this is the first carbon, or we say 1 prime. This is the second carbon, or 2 prime. This is the third carbon, or 3 prime. This is the fourth carbon, or 4 prime. And then the carbon up here we refer to as the 5 prime. So I always talk about that this is your, I'm writing upside down, sorry, high five. So if you think about giving somebody a high five, not right now because of corona, but if we're talking about the high five, okay, that is always going to be the carbon that is up and extended. Wow, that's a terrible carbon, or C. And then you have that phosphate group connected to that. So then when we talk about DNA and we talk about its directionality, okay, and I'm going to kind of erase and turn the board a little bit so I have more space. Okay, we talk about it being anti-parallel. So on the one side, you have this high five connected to the phosphate. And then underneath of it, you have another one where it's connected to the phosphate. And then it's kind of set up that this is the directionality of this side of DNA. And so again, if I count the carbons, you've got the 5 prime carbon up here. It's going to connect down here at the 3 prime carbon. This is the 5 prime carbon, 3 prime carbon, all the way down to, again, having the 3 prime at the bottom. So the directionality of this side, if I go down, is going from 5 prime to 3 prime. The anti-parallel portion of that is the fact that the other side is facing the opposite way. So Mr. Suter talked about the oxygen being on pointing down for this one. So then this is still the high five, it's just going the opposite way. So the phosphate's here. So this is actually 5 prime that's going to connect to the next phosphate molecule, or the next portion continue. So at the bottom of this one, and sorry, I'm doing upside down, you're actually going to have the 5 prime at the bottom and 3 prime at the top. So if you look at the directionality, on one side, you have 5 prime at the top, down to 3 prime at the bottom, and the other side is 3 prime at the top, down to 5 prime at the bottom. And it's going to connect in between with those nitrogen bases with a hydrogen bond. Okay, so that's quick talking about the carbon number and how we get that, and then the directionality a little bit more with the anti-parallel. Okay, questions about that before we move on? Okay, and then I'm switching back over to Mr. Suter.
All right, so Mike is now on. So real quick, before we start applying that, just looking over here at the room, we have 50 people watching. I love it, guys. Thank you so much. It means a lot, because I look at you as trailblazers, right? You're making history here. Whether you realize it or not, someday you're going to have your grandkid on your knee and telling them how you were doing bio in a live stream on the second day of the coronavirus epidemic. Anyway, Hannah Church, Esteban, Jake, Dalton, Evan, Mark Baker, Cody Supa, Daniel Corbin, Jessica, Andres, Kat, Elijah, Hannah Church, Alexi. And, I'm sorry if I'm skipping like guinea pigs for days. Daniel Corbin, wow, cool. If I'm skipping some of your names that we don't know who you are, maybe you ought to type in the chat who you are so we know. Anyway, quick shout out to all of those. If I missed anybody, I really apologize, but quick shout out to everybody that's here and hanging out with us. We appreciate that. So let's take this a step further and let's start talking about what replication is. So replication is really important because you have to copy your DNA, and your DNA is, like Mrs. Smith talked about yesterday, is the instructions for everything. And so how do we get an exact copy of that every time? Because if we mess that copy up, we're in real trouble, right? So that's the idea of replication here. We're going to replicate. We're going to make an exact copy of DNA. And so to show you this, I need to change my camera. So just hold on one quick second. We're going to go to a document cam. All right. So here we have a document cam. We have in front of us a diagram of replication. And so look at this kind of closely. I can actually make that full screen too. So there you go. So you can see a little better. what Mrs. Smith was just talking about, this 5' to 3' thing, is so important because it will dictate kind of how replication happens in some regards. And so pointing out a couple things to you, and I don't know if I'm doing this the way I would do it if we were in class, but hopefully it will make sense. On this diagram, we see the leading strand. And if we come down here, it says lagging strand. And because we see those two things, we can figure out which side of the DNA is running 3 to 5 and which side's running 5 to 3. We can figure out that directionality. So we know, now Mrs. Smith, if I get confused in the pressure of a live stream here, you'll jump in and correct me. But we know that the DNA template for the leading strand goes 3 to 5. Right? So if I come up here, this is my leading strand. It says it right here. Here's the template. That should be 3' here and 5' here. Looking for a nod from Mrs. Smith. Just to make sure. All right, so then we put that down. The leading strand, we add DNA from 5 to 3. What's interesting about that is that we can that happens all the time, right? So if we look at the lagging strand, if we come down at the bottom, well, if this strand is running 3 to 5 here, we know, since Mrs. Smith just told us about anti-parallel, this is going the opposite way, which means we're laying that down from 5. I don't know if you can see that 5 where I put it. 5 to 3. So that's a problem. All right, because we just said that in the beginning. Now let's figure out why that's a problem. We have this wonderful little, I stole these colored pens from my wife. Hope you appreciate it. We have this wonderful enzyme here called helicase. And what helicase does is it unzips the DNA. So you see helicase coming in and unzipping this DNA. If we had a better picture, we would see the DNA actually kind of unwinding right before the helicase gets there. Helicase comes in and is unzipping it. We're unzipping it in this direction. So this DNA polymerase come up here. So now we're talking enzymes. How do we know we're talking enzymes? Because they end in ASE. DNA polymerase, this is an enzyme that's adding free nucleotides. So we see our free nucleotides floating around. It's adding them. It's running right down the DNA. As it unzips, it keeps adding. But where's the problem? The problem is that we can't run down and follow the unzipping on this side. So this side has to go the opposite direction. So we have to wait for it to unzip a little bit and then start laying it down. Then it's going to unzip more and start laying more down. And then it's going to unzip more and start laying more down. So that becomes our lagging strand. It takes longer to build that strand. And let me flip pictures here. This picture maybe is a little harder to see, but hopefully you can make it out. What happens is, as the DNA keeps unzipping, this leading strand just keeps forming, but we get these pieces of DNA down here. And what actually happens, we didn't mention on the other picture, is each time more unzips, we lay down this RNA primer, which isn't really all that important for us to know about right now. But we lay that down, and then we make a piece. And we lay that down again and make a piece. And we lay that down and make a piece. And what we get are these fragments. And we have a special name for them, and I'm sure you've heard the name maybe already, which is the Okazaki fragment. Some people I know have already experienced that. So why are they called Okazaki fragments? Anybody know in the chat? Why are they called Okazaki fragments? Anybody got that far? Where did the name Okazaki come from? Because you would think they could have named it something a little bit better than Okazaki. I'll give it 30 seconds here. Anybody know why we call these fragments down here Okazaki fragments? Donald Trump, you're right. It is. I think it is Japanese. So why did we use that Japanese name? I'll give it to you. I'm not going to wait too long. It is a name. It's the name of the guy that figured it out. So if Mrs. Smith had figured this out, these would be Smith fragments, right? That's all it is. So don't let that Okazaki word scare you. It's just the guy's name. Anyway, that's pretty much it. If you think about it, What are the big things that you need to take away from this? Well, you need to take this guy away with you, helicase. Now, this is a DNA helix, right? So where did they get this name? They took the ASE, that means enzyme, and they tacked it onto helix, so they have helicase. We have DNA polymerase, which is the enzyme that we are using to add. They show it here in a couple different forms, which is the enzyme that we're using to add new nucleotides. Why is it called polymerase? Well, what are we making here? We're making a polymer. So we're making a polymer, put ACE at the end to get the enzyme. So taking away the enzymes, taking away the leading and lagging strand. And these pictures that I'm using right here, I posted a link to those in Moodle. So if you want to look at these later when you're doing something else, you can. And then one last thing real quick. What are we doing on time? Doing great. One last thing. We have, DNA is a huge molecule. If you took the DNA in your cell and you put it, sorry, I got to get this lined up under the camera, right? If you put it end to end, stretch it out, it's a huge molecule. It's really long. So if we started at one end and just went the whole way to the other end, it would take forever. So what happens is we get these bubbles, right? And then the bubbles go both ways. So what we just showed you, I want you to think about this. Look at this picture. All right, now look at this. What I just showed you is right here. We just showed you one side of it. So really, if you think about it, when the bubble opens, you get a leading and a lagging strand going both ways, right? And then those bubbles are going to meet and form a full piece of DNA. Another thing we talked about was the idea of this being, oh, that marker doesn't work. Let's see if we can find one that works. The idea of this being semi-conservative. Right? So why is this semi-conservative? So if you look at this final piece of DNA right here, we have an original piece of DNA right here, an original piece of DNA right here, and then a new piece and a new piece. So each new piece of DNA, think about it, break it down. Semi means half, conservative means to save. So why is this half saved DNA? Because each new piece of DNA contains half the original piece of DNA. And that's a great way to make sure every new piece is exactly what it should be. One last little picture here I had, just showing you that idea with the numbers and stuff on it. So if you were kind of confused about the 3', 5', and where everything is, you could pull this up on the link I put in Moodle. And I'll give that link to Mrs. Smith and check that out. All right, so I think, let me get rid of this document can. And guys, we're going through this quickly because you can come back and watch it again if you want. And we have some stuff to kind of follow up on that too. So in Moodle for today, there's only one other thing. And that one other thing is called a replication worksheet. And it basically asks you if you can tell me what the other side of the DNA looks like, which is the one thing that I don't think Mrs. Smith or I really talked about. But yesterday we talked about rules for matching nucleotides. So in the chat, we were going to do this, right? In the chat, can you tell us what the rules are? So if I have an A and a T and a C on the template side, what is the new side going to look like? Don't tell me that. Just tell me the rules. Haley Funk, A to T go together and G to C go together. Right. A and T spell at, and if G and C were sitting on the couch watching Netflix, they would be spooning. Which is a terrible, but anyway. So yeah, when you get off of here in Moodle, there is a link to a replication worksheet. Tomorrow in the live chat, we're going to talk about, maybe go over that replication worksheet and make sure that you understand that. Mrs. Smith, what else do you have? Do we have anything else?
I was just going to say, there's a lot of comments in there that they're confused. And I tried to ask, like, do you have any specific questions? I tried to go through and was typing as you were talking. Remember that we have office hours. Mr. Suter, yours is at 1 o'clock, I think. Right. And then mine was at 2. And I would be happy to, you know what I mean? Like if anybody that's Mr. Suter's students can't make his 1 o'clock and wants to do 2 o'clock, like with me, I can give them that link and you can, you know what I mean? You can hop on whether you're my student or Mr. Suter students, and Mr. Suter or my students, if 1 o'clock works better,
Yeah.
I can post that in there, and I'll put like both of our links to office hours in there. And it's this setup. Well, actually, no, I think I'm using Zoom. I don't know what you're using.
Yeah, we're using Zoom, but it's a video conference thing. You don't have to have your camera on.
No.
But if you want to come in, we can share our screen. We can share, we can run it through with you individually. Like I said yesterday, I get on at 1 o'clock. If I'm there by myself for 5 or 10 minutes, I'm getting off.
Yep, and mine ends right after my AP conference. So if I'm a minute or two late, just hang tight, because I might be finishing up with them if they had questions. And then the same thing, if I'm in there for 10 to 15 minutes, and nothing's happening, then, I mean, feel free to email me, and, we can do that. And yes, whoever is talking about RNA, yes, but we're not there yet. We're just talking about DNA replication. We're making more DNA because cells are going to duplicate. We're going to make new cells and more DNA that is exactly the same. We're not talking about RNA yet. So the questions that you have in there shouldn't be talking about RNA. We're only talking about DNA. And that's a big thing that gets confusing, is that people try to talk about DNA replication and transcription together, and they talk about RNA. We're not to RNA yet. We're only talking about DNA.
So back to that help session, you are free to email us. We will try to respond, but it is so much easier to explain this stuff in the help session, the office hours, than it is to respond. Sometimes, figure if I have 100 students email me wanting me to explain something or even 5, versus showing up and letting me help you at the same time, more of a small group kind of thing. So we can do that. Anything else? Definitely tomorrow at 10:30. 10:30 tomorrow. We'll be here. We're gonna kind of review this stuff a little bit. You'll notice there's no quiz on replication because we don't think you're ready for it yet. Yeah, it's a little confusing. We hit it quickly.
Yep.
But we'll hit it again a little bit tomorrow. We're going to go over, you're going to do the little worksheet thing today, which I think you'll find pretty easy. It's not overwhelming. I bet you if you give us 10 more minutes after you hang up on this, please reach out to your friends. We have 51 people watching right now. I'd like to see 100 tomorrow.
That'd be awesome.
I'd like to see 200 tomorrow. Because I think we have somewhere around 200 kids, right? Between the two of us?
Yeah, I have about 100. You have about 100. Does that make sense?
Right. We'd like to see 200. Where's the rest of our friends? Get the rest of our friends in here so we can do this together, and we're not having people make it up and struggle with it on their own. Let them get the help while they can.
All right. See you tomorrow.
Have a wonderful day, guys.